In today's video, we're going to be taking a look at mitosis for AS biology. So this is the mitotic cell cycle. We have mitosis, cytokinesis, and then this green area is called interphase. Now, interphase is made up of three different stages. We have growth phase 1, where a lot of protein synthesis occurs. Okay, there's a lot of mRNA, a lot of polypeptide synthesis is occurring. In S phase, DNA is replicated. Okay. And then in growth phase 2, there is further protein synthesis for the cell to continue growing. And also something that's quite important is that the pair of centrioles are replicated. Okay, so that we then have two pairs of centrioles. Now this here is the structure of a chromosome. We have two sister chromatids and we have a centromere which holds them together. The telomeres are at the tips of each chromatid. And the telomeres are repeats of a sequence of nucleotides which doesn't code for anything. Now the reason that why they are there is because in DNA replication, the enzyme cannot go to the very end of the DNA molecule. So some genetic information, some DNA is not replicated. Okay? And the telomere is there to prevent the loss of any important genetic information. Okay? Because the enzyme continues and it goes into the telomere, and because the telomere is like an extra, so we have our, if we're to think about it, if we have our double helix, right, and then we have this telomere, okay, the enzyme will continue. If the telomere wasn't there, it may stop here and important genetic information is lost. But because the telomere is there, it may stop up here, okay, and only some of that telomere is lost. So that's why telomeres are there, right? To prevent the loss of important genetic information during DNA replication. Now, moving on to the stages of mitosis. So the acronym that I use is PMAT, okay? Which means prophase, metaphase, anaphase, and telophase. Okay, now what happens in prophase? So in prophase, the DNA condenses into chromosomes, as we have here. The nucleus, the nuclear envelope disassembles and the nucleolus disappears. So what we're going to have at prophase are chromosomes free, right, within the cytoplasm. Okay, so they're going to be, there's not going to be any nuclear envelope, they're going to be within the cytoplasm with all the other organelles. What also occurs in prophase is that the pair of centrioles, right, and now these perpendicular structures, the pair of centrioles move to opposite poles and they start forming spindle fibers, okay, which attach as well to the centromeres of the chromosomes. Okay, now moving on to metaphase. What happens in metaphase is the spindle fibers arrange the chromosomes at the cell equator. Okay, so our chromosomes become arranged at the cell equator. They become aligned at the cell equator with that, due to that spindle, the spindle fibers. Now the spindle fibers span across the whole cell. Okay, and then during anaphase, what happens is the sister chromatids are separated from one another and then pulled to opposite poles. And what ends up happening is we're going to have these U-shaped chromatids being pulled apart from one another. Okay, as the sister chromatids are being pulled apart from one another, they're going to have this U shape. Okay. We also have our spindle fibers like that. All right, so that's anaphase. Now in telophase, the sister chromatids reach the opposite poles, the nuclear envelope reassembles, and the nucleolus reforms, okay? It reappears. So then what we're going to have is within our initial parent cell, right, we're now going to have two nuclei with genetically identical DNA. And what happens then is we then have cytokinesis, which means the splitting of the cytoplasm basically. So what's going to happen is down here on our metaphase plate, as we head up here, right, at the cell equator, a cleavage furrow will occur and the cytoplasm will begin to pinch. And what that's going to do, it's going to pinch, 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 and then two new daughter cells that are genetically identical will be formed. They'll be genetically identical if there has been no mutation, right? If there was a genetic mutation, then they will possibly not be genetically identical. Otherwise, they are genetically identical. Note, they are genetically identical. Because they're genetically identical, it doesn't necessarily mean that they are completely identical, because some organelles within the cells may not be evenly distributed, right? One half of the cell may have more organelles than the other half, right, than the other side. And so when cytokinesis occurs, one of the daughter cells may have more organelles than the other, okay? So that's really it for mitosis in animal cells. If you found this video interesting, take a look at some of the other content on my YouTube channel that might interest you. Otherwise, thank you very much for watching, and I hope you have a great day.